DNA goes in, a protein comes out. Watch one strand travel through the whole machine. It starts as raw DNA, just a long string in a 4-letter alphabet. First stop, transcription. Every T becomes a U and the DNA turns into RNA. Next, the RNA is read 3 letters at a time. Each triplet is a codon. Each codon is looked up in the genetic code and added as an amino acid. The protein grows. And out the other end, a finished protein, a working machine from a string of letters. Now, let's actually code it. Transcription is one line. Every T becomes a U, turning the DNA into RNA. Translation walks the RNA 3 letters at a time, looks each codon up in the table and appends the amino acid until a stop. And decode finds the open reading frame, the one that starts with a start codon. From a string of letters, a protein. Here is the problem. A real genome does not tell you where a gene begins. You can read the very same RNA in 3 frames, shifted by 0, 1, or 2 letters. Each gives a different stream of codons. But only one of them opens with AUG, the start codon, and runs cleanly to a stop. That is the suspect. That frame is the open reading frame, the actual gene. Translate it, and out comes the protein. Now let's break it. Take one codon, GAG, glutamate. Watch what one flip base does. Flip the 3rd base and the codon still codes for glutamate. Nothing changes. We call that silent. Flip the 1st base instead and you get a stop codon. The protein is cut short. That is a nonsense mutation. But flip the middle base and glutamate becomes valine. This single change creates the sickle-cell variant of beta-globin. And the worst kind, insert or delete a single base and every codon downstream of it rereads as garbage. A frame shift. But a real genome does not hand you the gene. It is buried in a long strand of mostly nothing. So we scan for every start codon, ADG, and read each one to the next stop. Those stretches are open reading frames. The longest open reading frame is the strongest candidate for a gene. Translate it, and there is your protein. So that is the whole machine. Transcribe, translate, find the reading frame, and see what one mutation does. A gene, decoded from a string of letters,